Hi, my name is Jailin and I'm going to talk about transformation. Transformation is a process in which bacteria take a foreign DNA that is free in the environment, not enclosed in a cell or virus, and incorporate it into their own genome. This allows bacteria to acquire new genetic traits such as antibiotic resistance or virulence factor, which can be beneficial for survival. The concept of transformation was first demonstrated by Frederick Griffin in 1928 in his famous experiment with Streptococcus. He observed that a non-virulent strain of bacteria could be transformed into a virulent strain when mixed with heat-killed virulent bacteria, suggesting that some transforming principle was responsible for transferring genetic information. Some key features of transformation are naked DNA transfer, unlike conjugation, which requires cell-to-cell contact, or transduction, which involves bacteriophage. Transformation involves the uptake of free DNA from the environment. Another one is the size of DNA transfer. Transformation involves that uptaking of about 20 genes, though this can vary independent of the bacteria species. DNA sensitivity. Since the DNA involved in transformation is free in the environment, it can be degraded by DNA, an enzyme that breaks down DNA. This is a distinguishing feature of transformation, as conjugation and transduction are not affected by DNAs. There are two main types of transformation. Natural transformation are some bacteria that have specialized proteins that enable them to take a foreign DNA naturally. Examples of naturally transformable bacteria include Streptococcus, Haemophilus, Neisseria, and Bacillus. These bacteria have competence factors, proteins that regulate their ability to take up DNA, competence that can be influenced by environmental factors such as nutrient availability and cell density. Artificial transformation is many bacteria that are not naturally competent but can be induced to take up DNA in a lab setting using specific techniques like chemical transformation. There are cells treated with calcium chloride and then exposed to heat shock, making the cell membrane more permeable to DNA. This method is commonly used in molecular biology to introduce plasmids into E. coli. Electroporation are another example of artificial transformation. A high-voltage electric pulse is used to create temporary pores in the bacterial membrane, allowing DNA to enter. This method is more efficient than chemical transformation and is used for both bacteria. The significance of transformation are like genetic diversity. Transformation allows bacteria to acquire new genes, which can lead to increased adaptability and survival in changing environments. Antibiotic resistance are some bacteria acquire resistance genes through transformation, contributing to the spread of antibiotic resistance in bacterial populations. Biotechnology applications are transformation that is widely used in genetic engineering, such as cloning and the production of recombinant proteins. Examples like insulin production using genetically modified E. coli. Bacterial transformation is a crucial mechanism of horizontal gene transfer that allows bacteria to acquire new genetic traits, whether occurring naturally or artificially in a lab. Transformation plays an essential role in bacterial evolution, antibiotic resistance, and modern biotechnology applications.
Transduction is another method of horizontal gene transfer in which bacteriophages, which are viruses that infect bacteria, transport genetic material from one bacterium to another. Transduction happens when a bacteriophage infects a bacterium and accidentally incorporates bits of the bacterial DNA into its own viral DNA, and then transports this bacterial DNA to a new host cell. There are two different types of transduction. There's generalized transduction. There's specialized transduction. Generalized transduction means that any section of the bacterial genome may be transferred. That occurs during the lytic cycle of a bacteriophage. Specialized transduction means only certain parts of the bacterial genome can be transferred, and that happens during the lysogenic cycle. And the role of transduction in bacterial evolution is that it facilitates the spread of genes responsible for antibiotic resistance and metabolical functions across different bacteria. So thank you.
Conjugation is the process where two organisms, one donor and one recipient bacterial cell, transfer genetic material to each other through direct contact. So, an example would be F plasmids from E. coli. It's important because conjugation helps bacterial cells spread antibiotic resistance to one another, which increases genetic diversity and adaptability. It'll mediate the propagation of metabolic properties, so like symbiotic lifestyle, virulence, biofilm formation, resistance to heavy metals, but the most important one would be antibiotic resistance. The conjugative process starts with a donor establishing direct contact with the recipient cell through a sex pilus. So it's like a hair-like appendage that's just on the side of it, and it makes contact with the recipient. And after they make contact, it pulls the two cells together. So it just like contracts. And then once they're pulled together, a single strand of DNA is then transferred from the donor cell to the recipient and then replicated in both cells. And after the old donor contracts back, the recipient cell becomes a new donor with the new information. It's just important for like spreading that antibiotic resistance, like spreading that one single piece of information, and then it's replicated in the old cell. And then since that cell is now a new donor, it can go and replicate that information in a ton of other cells too. So that's why it's important.
Hello, my name is Jennifer, and today we're going to be talking about transposons. It's a piece of our genetic code, and it's usually referred to as jumping genes. They are essentially DNA sequences or stretches of DNA, and they have this unique ability to relocate within the genome itself. And like I mentioned before, they have this nickname called the jumping gene, which kind of captures the idea of them hopping from one place to another. Some use a cut and paste mechanism, while others use a copy and paste mechanism. This movement can have a variety of effects, some being good, some being bad. One major impact being that they can disrupt the genes itself. If a transposon jumps into the middle of a gene, it can interfere with its function. This can then lead to mutations or even a disease. However, it's not always negative. They can also lead to genetic variation. This variation is essential, allowing populations to adapt to changing environments. Now for the second slide, we're going to start talking about different classes and different types of transposons. So to start off with our first class, I mentioned it before, this class is class number 1, and it basically just works by copying itself and doesn't move the original at all. First, its DNA is turned into RNA. Then an enzyme called reverse transcriptase converts the RNA back into DNA. And this new DNA copy is inserted into a new location, and the original will stay put. Now for class 2, which I also mentioned before, it uses more of a cut and paste technique. It's just how it sounds. They physically cut themselves out and put themselves in a new location. They do this by using an enzyme that's called transposase. This allows the DNA transposons to be cut out and inserted somewhere else. And this basically concludes the overview of transposons. Obviously there's a lot more to it, but this was just a really quick overview.
Our plasmid, what is it? Bacteria have a small piece of DNA called plasmid, which separate from their main DNA. It's like a little extra set of instruction for the bacteria. Our plasmid is a specific plasmid that has a gene that help bacteria survive from antibiotics. Their structures has a 80 to 95 kilobytes length containing phenol gene, which helps control the transfer of the plasmid between bacteria, and has a circular DNA molecule. What are our plasmid functions? So the functions help bacteria spread drug resistance. They carry genes for replication, sharing genes, resist ampicillin, and help bacteria resist antibiotics, and grow pili for gene transfer. This means resistant plasmid can make copies of themselves without needing the bacterial chromosome. This allows the plasmids to duplicate and be passed on to other bacteria during cell division. Mechanism of resistance. Bacteria can protect themselves from antibiotic in several ways. They may block the antibiotic from entering their cells by changing or reducing the number of entry points. Some bacteria use pumps on their cell surface to push the antibiotic out. Others produce proteins that break down the antibiotics, rendering them inactive. Additionally, bacteria can alter the part of their cell that the antibiotics target, making the drug ineffective. What is horizontal gene transfer? Horizontal gene transfer is a process by which bacteria share genetic material without reproduction. This allows them to gain new traits such as antibiotic resistance. Examples of bacteria that carry our plasmid. One here is E. coli, Staphylococcus aureus, Pseudomonas aeruginosa.
Hi, my name is Rebecca. I did the slides on genomic islands. So the definition are genomic islands are the regions of DNA that have been acquired from other organisms through lateral gene transfer. They are the key dividers of microbial evolution and the adaptations of sharing the landscape of the life of the earth. Some examples of the islands that we have are pathogenic islands, which are specific islands that carry valence genes such as those are found in Salmonella. A symbiotic island are islands that allow bacteria to establish a symbiotic relationship with its hosts. And then genomic islands in the lateral gene transfer. Horizontal gene transfer are genomic islands that DNA have been transferred from one organism to the other, often through a process like conjunction, transduction, or transformation. Conjunction are basically like a cell-to-cell contact transfer. Transduction are a transfer of DNA of macrophages, which are basically viruses that infect bacteria. And transformation are an uptake of naked DNA from the environment. Some distinct features of these are they have a higher GC content, they have a different condon usage bias, and they are in the presence of micromobile genetic elements like transpones and indigenes. The importance of the genomic islands in the microbiological adaptations are antibiotic resistance. So basically, they're islands that resist antibiotics, allowing the bacteria to survive in the presence of different types of drugs. The significance is that it's a concern to people in healthcare, as it contributes to the spread of multi-drug bacteria resistance, and having drug resistance not really good. Some valence factors, they can also carry genes from valence factors allowing the bacteria to cause diseases. For example, the T3SS, which is a type 3 secretion system in the pathogenic bacterial facilities, and the injections of vulgence of proteins into the host cell. Some microbial diversity. The genomic islands can introduce new metabolic pathways, allowing the bacteria to adapt into different environments and utilize the novel resources there. This enables the bacteria to exploit a wide range of ecological niches, from extreme environments like the hot springs that you can normally find them in, or the human intestines. And then our Christian worldview perspective is from Acts 20:35, reading, "In everything I did, I showed you that by kind and hard work we must help the weak, and remembering of words Jesus himself said, it is more blessed to give than receive," meaning that God blesses who work hard for his glorification and for the betterment of others. This passage explains that we must help the weak in healthcare, which means in healthcare, we must help people who are struggling with all types of illnesses at all types of time so we can glorify God.